Hello, good afternoon. Welcome back to the MedAID final event. We are going to continue this afternoon. We are concentrated in what we call the zootechnical work packages. We are starting with a presentation of the MedAID dashboard. And then we continue with the updates and presentation of main results and recommendations from the work package 2 on zootechnics improvement, work package 3 on genetics, and work package 4 on health management and welfare. So we are starting with Cristóbal, please.
Okay. Hello, everybody. Good afternoon. Well, we are ready again. The dashboard, the MedAID dashboard, is a consequence of the integration of all the assessment that we are doing in the other technicals work packages, and how we can integrate it and propose solutions at a hard level. The approach that we do at the beginning is the idea to test the validity, the solutions, and industrial scale, no? Be the sustainability, implementing a specific and adult software. We know that existing actually in the market for the industry amounts an amazing quantity of softwares, platform, different kind of platform that integrate information, biological, technical, zootechnical, economical, and other in some more cases also they are integrated in an ERP model or whatever. But the idea that we have at the beginning is, I love data. We need data to try to understand what happened in the Mediterranean industry, to try to understand what are the tendencies, how do this one, etc. And also transforming all the data in value. When we ask to the companies we collaborate with us in the MedAID at the beginning 3 years ago, they normally ask to us that they oftenly receive many surveys from different kind of organisms, like governmental survey, sectoral agents, productions, European, etc. And in some cases, they are continually answering the same questions. And in most cases, the question that they are demanding is the same, but they are formulated by the different ways, a different form. And we know, we observe that in most of the cases, the information that coming from these surveys are incorrect or not correct answered. We found that inconsistency of the data, no harmonization. So, with that said, the challenge that we introduce is how we can manage of the data, how we can use the data that we are obtaining in the different kind of surveys to provide a strategic decision-making model using a single platform, integrating. Until now, until this moment, we are not found any specific platform that integrate the data as the model that we are using. I don't know if this exists, probably, but I can found, and I do a very deep search in the different models existing. To apply this one, we decide to generate a model, and the model that we apply is first of all try to understand which are the structure of the companies who are doing aquaculture in the Mediterranean, based on a technical approach and economical approach, using economy, production, and technology assist models. Why we do this one? To do this one, first of all, we need to know which kind of species they are producing. 1, 2, both, 3, more than one. Which is the activity, the main activity they are doing? Hatchery, growing, combining one species to the other. Later on, which is the level of the degree of technology that they are applying. Okay, high super specialization in producing only one species, or combine some, etc. And this is the model that we are structured to assign the characterization of each specific company. So asking some question, minor question like what species you are producing, what are the size of the company, do you have, how many tons, how many million fingerlings, you are integrated or not. We proceed of the data and we provide at the company the model of a structure, organizational structure that they belong. So we use this one to help and understand difference between facilities, because at the end, also that we use data at the company level, we decide to apply at the facility level. Why? Because in some cases, facilities are isolated one to the other, maybe in different countries, on different cities or whatever. And we offer the possibility to know which are the behavior of these specific facilities inside to the company. Well, the idea is to help to understand differences. It's based in annual production and species differentiation. So the companies can obtain information about year by year. Till now, we don't have a temporary sequences, maybe in the future, analyze all the years together. Now, the specific analysis is by year, and also you can select it by species, okay? Also, we are including some auto-evaluation auditory models to help the companies to observe which things they are doing in different approaches later on. And also the idea is to help in a process of benchmarking in general, using the data, aggregated data at the company level. Well, they are data. In some cases, it's some incongruences, but you can use this if you like it, but with a particular approach by each facility. I think this is quite specific. So we offer and we help to facilitate to add or to introduce some potential improvement based on the data coming from the average of the sector. So you have your data, you know your data, and you can observe which is the behavior of your company, your facility, with the average of the sector. So you can decide if you are or not in condition to take some strategic decision. And finally, all the data, of course, is processes and totally anonymous, and is protected absolutely, following the specific data protection, etc., etc. And well, it's no problem. One of the important things on this model is we don't provide to the company or to the facilities the concept of you are doing well the things or not. No, this is a decision of the company. We say, this is the data you are provided us, and this is the average of the sector. You can decide if you can improve or not, or you have some indexes to, okay, to do something, but this is your decision. We don't define the quality of the data. Actually, we are integrated all these MedAID platform in a platform that similar to the first that we are presenting, which is a Smart Water Planet Suite, which is a platform building. So we are not created a new one. We are using ones that existing, and they have enough information, enough structure to support the needs coming from the MedAID. In this platform, we are integrated in a specific suite of the MedAID, and actually we are adding intercomparative models based on the work package, or the technical approach, okay? Zootechnics are available, health and biosecurity, economics, environment, welfare, and governance. And now we are working, might be in the future, to integrate in genetics or product, which kind of product we are doing. This is a decision that may be coming from the sector. If they have interest or whatever, we try to do it. And okay, I got to change. Yes. Well, sorry. We have two important things today. Actually, this platform is available for whatever. It's free. It's free. A change of the data. If you provide data us, we offer you the possibility to go inside, introduce the data to the intercomparative model. Okay, almost during the I think the first 2 years, we have the possibility to offer this characteristic consequence. The first step is go to the smartwater.com setting and introduce your data. You register ICA users, you introduce some data, and they assign you the possibility to go inside to the benchmarking comparative model. Okay, once you are in the entrance, we demand use some data like the name of the company, which is a model that I can generated, my company, IRTA, okay, to test and validate all the data and be sure that all is working well. Actually, we have 34 different companies and users using this platform. Which is the model of the production sector that you have, which are the user, etc. And it's the you are under protection data, you iterate confidentiality, etc. And you provide data. Once you are introduced that, you go directly to the Smart Water Planet platform in MedAID dashboard. In this, you have the data entry. No, sorry. First of all, you need to configure your company. And with the configuration of your company, adding the different kind of facilities that you have, okay, we are generating different facilities that all are integrated in my own company. We aggregate the category. So this is the category that you use to do the benchmarking, intercomparative benchmarking, if you like, with companies or facilities like you. Okay, you can compare at 2 levels. Based on the region, which is the activity, etc. And we have some things about how many millions you produce, which are your tons, etc., etc. The locations to be sure that is connected. Okay, once you are introducing the data of the company, you have different models. In the models, we are doing as questions. In some cases, it's a drop list. In drop list that you decide, that you chose, which is the answer. Basically, the model that we apply to the auto evaluation tests or auditories. And in other cases, we ask some very elemental questions like, for now, the hatchery, which is the survival, which is the average of deformities, which is the growth, which is the TCR, etc., etc. And in every case, we have a glossary. The glossary is a document who explains exactly which is the data that we are demanding and how it's necessary to calculate it. In some cases like this is so simple. Survival at day 30 is the survival that you obtain by dividing the number of fish alive at day 13 per hatch by the number of the larvae hatched. Quite easy, but this is standardized the how we can obtain the data. But in other cases, like economics, I think, yes, our college from University of Cantabria, they are working on and developing the model, introduce R of the model, which is the system to calculate, for example, other production costs. This is a complete description of the methodology that we are using. This is published in the paper, etc., that we are generated in during the MedAID, or which is the labor cost, which is the money to obtain, which is the energy, etc. On also how we can calculate. Sorry, is an example. Okay, which is the model that we use to calculate the operational profit margin. The operational profit margin is based on the calculation of EBIT, and to calculate this one, you need to follow this formula. So it's to facilitate it as much as possible. Okay, once we are introducing all the data, the next is the dashboard. In the dashboard at the beginning, you say, did you remember? We have introduced the data year by year. Okay, this is the first overview that the company have, and they say, okay, you are introducing all the data that we are demanding you to do the analysis in all the different modules for the 2018. It's okay, but if, for example, I check the data that we have in 2020, I say, okay, we are introducing data in zootech, in welfare and environment, but we are not introducing some, we don't do the auto-evaluation on health and security, on economics and the governance. Okay, you need to introduce more data. It's available to you. As much data we have, more powerful is the system to help you to do the inter-comparative or benchmarking model. Oh, one of the information that we present is quite easy, is quite responsive, is very, okay. So the first one is the presentation of, for example, labor costs, which is the percentage of each one, and some cases of the impact of the climatic change in your company, which are the operational margin that you have, and also the biomass that you are producing. This information is absolutely, is the information that the company who introduced the data are seeing. It's not for other one. The unique information that we have is, the unique information that we are sharing is the average of the mode coming from the data that we are introducing. Like, for example, welfare. In welfare, we are, dashboard. In welfare, we are introducing, dashboard. Welfare. Okay, in welfare, we are introducing auto evaluation test, test, including 12 or 13 principal indicators coming from the analysis of operational welfare indicators doing in the work package. I don't remember in the MedAID. We are introducing this indicator, and you are asking. When you are asking to these questions, we offer you the possibility to use this signal, which is your actual level of your company following the indicator that we are introducing compared with the whole sector. Okay, but if you like, you can select it by companies. And they say, okay, your company, Interfingerlings, belong to super specialist mode. So this is the comparison between you and the facilities are exactly the same level like you. On right, you have which is the mode of the answering coming from the sector. So you can help, or you can use this one to take decisions. Is, for example, you have, I don't know, minus, I don't remember now. Okay, in these cases, you say, I have a terrible company. We are doing nothing in welfare. I know the answer for the sector, and I can take a specific or strategic decision to improve this one. Okay, and we are doing more of them. Finally. Yes, okay. So, okay, finally, in brief, the benchmarking is a two-facet, I would say, in annual production, differentiation by species, self-evaluate model to help the industry at the company level. Okay, they are also introduced using smart water cloud because it's possible in the future to introduce transactionality of the data. In the future, we can exploit this one using an agreement, public-private, with this company to maintain the data in the future and using and add value and introducing new species, etc., etc. And because both platforms are interconnected. And well, we invite you go to with the platform. You might be our company or not, because if you are some stakeholder, you can introduce some information, you can generate it your company. This is one, later on we clean, to be sure that the platform offering you the possibility to intercompare, etc., etc. And improve continuously. This is the idea. And also maybe in the future, our vision is this platform will be used for all the governmental or stakeholder or whatever to ask only one time for each company the data that they require and sharing in a common.
Thank you. Let's continue. We move to the work package 2. So Alicia, please. A ver, ¿dónde ha puesto este hombre? Espera, ¿qué es? ¿Dónde lo ha puesto aquí?
Porque no sé dónde está.
Es que está en uno que se llama...
Y para pasar es como siempre, ¿no? Le doy la flechita para arriba y para abajo.
Aquí se ve entero. Bueno, ¿quieres puntero o no?
No. Vale. Ah, vale, le voy pasando aquí. Ok. Está... Sí. Okay, thank you. Thank you, Bernardo, and thank you, Cristóbal. Okay, I'm going to talk about the work package 2 that has nothing in common with the others because we were mostly focused on the sunset, so technical problems that the companies were already detected during the production of seabass and seabream in the Mediterranean. So the objectives were, The objectives were to address the problem of fat deposition in seabass especially, improve the quality of the juvenile by means of epigenetics, determine the optimal conditions for juveniles to apply protocols for improving their performance, reduce the conversion rate, improve health and stress tolerance, using better rearing strategies, and improve the welfare by minimizing the stressful events through nutritional modulation and immune stimulation. So these were the objectives that we proposed in the project. And the tasks that we selected were these ones that I'm going to introduce you. So for the objective 1 is to address the problem of fat deposition from a multidisciplinary approach. The first task was to assess the feeding strategies to reduce the adipose tissue. This was carried out by Yanis Kotsamanis in HCMR in Greece. And what he did was to have the seabass from 12 grams until almost commercial size for 170 days of culture. He separated lean fish and fat fish regarding the weight, the final weight that they had. And what they did was to check what were the conditions of the fish and see the difference between lean and fat fish. What they found was that when you finish the growing period and start measuring the different somatic index, the only one that gives good information about the fat accumulation in the seabass is the perivisceral fat index. The hepatosomatic index was found not to be related with the fat accumulation, and only visceral, and even the condition factor neither. Only the perivisceral fat index was a real one, the one that gives you the good information about the fat accumulation in seabass. They also studied the effects of the effects or several hormones, growth hormone, ghrelin and leptin, that are related with hunger. They studied if there was any relationship between this hormone production by the fish and the fat accumulation, and they found there were no relationship between the hormone production and the fat accumulation by seabass. They will still continue working with this because they couldn't find any factor involved in this fat accumulation in the visceral fat. Okay. The second task was to check what kind of feed composition and temperature are, what kind of feed is much appropriate when you change the temperature between winter and spring and autumn and winter, what kind of fish use or feed used to avoid also the adipose tissue accumulation in the seabass and seabream. The trials were carried out in two places. With seabass, it was in the HCMR with Stavros Chatikotis, and seabream was carried out in the University of Bologna by Alessio Bonaldo. What they used was two kinds of feeds. Usually in the project, we use commercial feeds already in the market. We were not formulating new feeds. So we use a feed composition, with 43% of protein and 16% of lipids, and another one with a little bit higher content of lipid, and feed the fish with these two feeds and at 17 degrees, then change to 23, and also change the feed from low lipid to high lipid, and check what happened with the fat accumulation. And what we found was that any kind of feed can be used during temperature changes because there were not exactly an effect. Only it seems that the low dietary lipid level seems more appropriate for seabream, but in the case of seabass, any kind of feed will have the same effect during these temperature changes on fat accumulation in the seabass. So for seabass, there was no effect. For seabream, a little bit. If you use a lower dietary lipid level, it is much better for the seabream. The second objective was to improve the seed juveniles for growing purposes, and this was carried out in the HCMR by Elena Sarropoulou and Constantinos Mylonas. And the subtax was just using, he will explain later much in detail, but it was using 3 different temperatures, 15, 17.5, and 20 degrees, to carry out all the larval development and check later on what was the differences in growth, in sex differentiation, skeletal deformities, different aspects of the physiology of the fish. But Tinos will present much more detail about the results of this task. The third task also was to determine the optimal condition. The third objective was to determine the optimal conditions for juvenile to design and apply protocols for improving their performance. And one of the first tasks we did with this objective was to check the water current conditions that were better for the fish to develop swimming behavior. This was carried out by Arjan Palstra in the DLO and by Ana Roque in IRTA. And they were checking seabream with a train to swim at two different speeds, 1 body length per second and 2 body length per second during 8 months, and then use those fish in sea cages. But he will explain in much more detail later after me. Another task in this same objective and is the same objective to check the quality, improve the quality of the juveniles during the rearing, was to check the fish density and daily feeding frequency. In the case of fish density, we did the experiment with seabream and seabass. Seabream was carried out in the University of Bologna, and seabass was carried out in Tunisia. And the work was using 2 different feeds and 2 different densities, 10 and 30 kilos per cubic meter, and different fish meal fish oil ratio, 35, 15, 15, 3, and check if there was any effect of the feed on the fish in high or low density. And what we found was the diets containing low levels of fish meal fish oil can be used with seabass and seabream when they are rare at very high density, from 36 to 44 kilos per meter cubic during the growing phase. So it's better to use low fish meal fish oil when you are rearing the density, or when you are having the fish in the sea cages at very high density. So this, I'm talking very fast, but anyway, you will find all the information in the deliverables. They are already uploaded in the web and in the different papers that we have already published. So all the information and all the detail of the experiments we have carried out are in the deliverables and in the publications. Again, with the same task, assessment of the rearing conditions to improve juvenile quality. One was fish density, the other was feeding frequency. In this case, what we did was to separate juveniles and on-growing fish and check what were the best feeding rates for different ages, ages of the fish. It was carried out in the University of Bologna from the group of Alessio Bonaldo for on-growing fish, and in the University of Turkey by Ali Kurkut using juveniles. In the case of juveniles, they were using the same feeds, fish meal, fish oil, 13, 15, 15, 3 that we were using in the trial with the fish density. It was exactly the same feed. But in the case, and they were trying 3 different doses, 1, 2, or 3 times per day of feed. provided even both to juvenile and to on-growing fish. In the case of the juveniles, there was an effect, a clear effect in growth of the fish and in all the aspects of performance, conversion, growth, etc., when they were fed 2 or 3 times per day. But in the case of on-growing fish, as everybody knows, there was no need for feeding so many times. Only 1 dose per day is perfect for the on-growing, and 1 dose was recommended. The last task was to optimize the feeding strategies used during unfavorable farming conditions. It was, again, everything is included in the deliverable 2.3. And one of the first tasks was to optimize feed formulations, especially during winter conditions that are the worst. So the experiment was carried out by HCMR, by Sofia and Grola, and they were using 2 different protein lipid content, and during the temperature in winter, 13 degrees. And they introduced another diet that was adding some supplementary additives, krill meal, beta, soy lecithin. So at the end, the best feed was that one with the lower also feed, fish meal, fish oil, and protein lipid content for the winter period. Okay, I continue. The other one was to use this feed formulation when you are using the fish in during crowding stress. Crowding stress is mostly used in the sea cages during grading, and also during how can I say, antibiotic treatments or antiparasitic treatments. So we selected this crowding stress to check if there was any effect of using different feeds for the recovery of the animals of the crowding stress. So we use high, medium, and low fish meal fish oil ratios. For 60 days, we ended the experiment and fasted the fish for 36 hours, and then submitted the fish to the crowding stress high density for 50 kilos per cubic meter for 2 hours. Then we took some samples of blood and mucus, and in both cases, low feed meal fish oil resulted in a lower final weight and a specific growth rate. But in the case of seabream, there was no effect of the feed on the crowding stress recovery. In the seabass, there was a little bit of effect in the skin mucus parameters, but in both cases, there was any kind of feed can be used during the crowding stress. Okay. Finally, we included another task with introducing new functional feeds to check the immunocompetence of the animals. This was carried out by Enrique Gisbert in IRTA, and we tested 2 kinds of additives. Was one spray-dried plasma included in the diets with very low fish meal level, only 5%. And the other one was using medicinal plants extracts. In the case of the spray-dried plasma, it looked that it was a very promising dietary alternative to be included in the diets with very low fish meal, because there was an increase in the immune resistance of the animals. In the case of the medicinal plant extracts, there was an enhance of the non-specific immune response, as in the case of the other feed additive, and this can be recommended for the incorporation in aqua feeds when you are going to as a prophylactic agent. Finally, I'm trying to go fast. What we did with all these results was to do a case study using 2 different sea cages in 2 different areas of the Mediterranean. There was a trial carried out in Galaxidi in Greece, and the other one in Cudamar in Alicante in Spain, using the same batch of fish, juveniles that came from Galaxidi. They were distributed in the 2 sea cages, both were the same, 60 meters of diameter. And what we selected was to use different digestible protein, digestible energy during winter conditions, and use only 1 feed dose, because there were some of the recommendation or the results that we got from the different experiments. We compared both sides, and the results were almost the same. There were no differences in the growth or in the conversion rate or in the survival or anything. And the good thing was that at the end of the growing period, the fish were of very high quality. We used them for a customer evaluation, and the results were very good and without any difference between the two locations. So that's it. At the end, I have added that also using this suggestion, this recommendation that we gave to the producers of using this low protein energy ratio during the winter and increase during the summer, the cost of the diet was 10% cheaper. So it was not under the scope of the work package, but we found that there was an economic profit of these ideas that we suggested. Okay. And just that all the results are presented in the deliverables. They are much more in detail that I have said now, and if you can check them and see. Okay.
Okay. Thank you. So next is Dinos. Okay. Go. Okay. Help me to find yours. Is Mylonas.
No.
Okay.
How do I start the presentation?
Yeah, like this.
I just go for
yeah.
Okay. Okay. Good afternoon, everybody. As Alicia said, we were in charge of a task on looking at the effect of temperature, early larval rearing temperature in seabass on growth, sex ratio, and deformities. And this was a continuing work that started many years ago with first the focus on the sex differentiation. And we just wanted to add more information on this equation of what are the aspects of different larval rearing temperature and what different systems in fish in seabass they affect in relation to aquaculture efficiency. So as the graph on the left shows, I cannot point very well from here. We examined 3 different temperatures that we knew from the past that will give us differences in sex ratio. This was the original experimental design. So 15, 17.5, and 20. We exposed the eggs until the stage of metamorphosis to this temperature. And then we kept a constant temperature throughout the next 2 years, and we grew fish up to harvest size. Then we took different samples at different developmental stages, and we monitored growth and different parameters that are shown here. Basically, we looked at growth to confirm, to know what is the effect of the temperature that we want to propose. Looking again at sex differentiation and sex ratio, because as you all know, there's a sexual dimorphism in growth in seabass, and females grow better, significantly better. So we want to have as high a female percentage as possible. We looked at skeletal deformities again, and we looked at the vision system and how the vision system develops. And then this was our first ever effort at looking at epigenetic effects during early development on different aspects. And unfortunately, we started in this project looking at this, and we have some very interesting results. But it's a very early stage, and we're looking forward to the future to start identifying what these epigenetic targets do in the fish. So very briefly, as we expected, the 17 degrees and 20 degrees gave much higher growth than 15 degrees, but high temperature reduces the the differentiation to female sex. Therefore, the optimal of the three temperatures, as we expected, was the hypothesis in the design. The best temperature was 17.5 degrees because it gave us the highest sex ratio, the highest female percentage, without affecting growth, which was exactly the same as the 20-degree temperature at harvest time. Looking at the effect of different temperatures in deformities, in the past we have seen that high temperatures have the propensity to produce more deformities, skeletal deformities. We didn't see this here, so apparently deformities depend mainly on other things than temperature, probably the water current and also the diet of the fish. So in this particular experiment, we did not see any temperature effect, and overall the deformities were very low. But again, we can see that 17 degrees did not have any negative effect here. Looking now at the vision system, and we looked at the two major components of the vision system, the cone cells and the rod cells. The cone cells are the ones that are used for color vision and during the day at high intensity. Rod vision is what you fish animals use for low intensity. And what we have seen is that at the too high temperatures, we had a higher number of cones and rods being developed at the stage at the stages that we looked at, especially metamorphosis. This means that the fish will be having a better eye at that time. So both the 17 and 20 degrees worked equally. So given the fact that at 17 degrees, the metabolic rate of the live material, of the live feed, is lower, we propose that of the two temperatures, it's better to keep the 17.5 because the fish will have the ability to feed on more nutritious live feed. Looking now, as I said, on the aspect of epigenetics, we looked at microRNAs. We looked at an array of different microRNAs, and we identified that there are significant differences in the presence of these mRNAs due to temperature. And we have seen that most of the temperature influence are during the flexion and the all fins stage. And we did identify distinct pathways that seem to be significantly enriched, the epigenetic modification of these pathways. This is a figure that I don't understand at all, so I'm not going to say much. Basically, these are the different pathways that we looked at, and you can see that because of the difference in temperatures, some of them are upregulated and some of them are downregulated. That means that temperature has the ability to epigenetically modify a lot of different aspects of the fish's physiology. And this is my last slide to conclude. So basically, with this evaluation, what we conclude and propose to the industry is that 17.5, so 17 degrees, is the most appropriate temperature for larval rearing of European sea bass because it provides higher female percentage. It provides optimal growth, similar to higher temperatures, no deformities. This is similar to all the other temperatures, better eye development than lower temperatures, and slower metabolization of nutrients by live food. In the epigenetic modification aspects using mRNAs, again, smaller microRNAs, we saw that there was a regulation of the immune system related genes depending on the temperature. So depending on the temperature, you can affect significant immunity genes epigenetically. And most of the mRNAs seem to be acting at the level of the stage of flexion and all fins. So this is the critical stage that these things happen. And of course, now it's our task to try to make sense of all these different microRNAs and the different pathways that they affect, which ones are upregulated, which ones are downregulated, what are the functions of this. But obviously, this was an important result that shows that the environment epigenetically can significantly affect a suite of physiological factors in this fish. So thank you very much.
Thank you. So, Arjan. No, you have the next one.
You can try. Good afternoon. My name is Arjan Palstra. I'm a senior researcher fish physiologist at Wageningen University and Research, the Animal Breeding and Genomics Department. And what I would like to present for you today is the studies that we did in MedAID in collaboration with IRTA, with Ana Roque, on the optimal flow for seabream, both during land-based rearing and then during the follow-up stage in the sea cage. So the objective was to design an exercise protocol for seabream in a RAS environment with a main objective to increase robustness of the fish and to evaluate also the effects of these exercise protocols during the on-growing in the sea cage period. And we tried to follow that, of course, because that's very difficult to follow activity patterns in the sea cage. Can be done by cameras. What we did was we monitored behavior and activity by using accelerometers, so sensors in the fish that can measure acceleration. So basically we did two long-term experiments. One was swimming for 8 months, either at 1 or 2 body lengths per second versus resting controls. And that was done for 8 months and follow up by a 6 weeks experiment in the sea cage where we performed the accelerometry. And there was a collaboration with Pablo Aura Lopez from IMEDEA. So what we found, first of all, was already a significant difference at 20 grams. Then the exercised fish at 2 body lengths per second already reached that stage, while the other ones were still behind, between 10 and 15 grams. And finally, after 8 months period, we see a significant growth effect, so swimming enhanced growth, swimming at 1 body length per second, so about 15%, and then swimming at 2 body lengths per second, about a doubling, 36%. But also very importantly, we see that the individual variation becomes much less, about 25% less. What we did after the exercise experiment was a stress challenge in order to see if there were any differences there. What we found was a difference in the basic cortisol levels, but not during peak levels or later after recovery. So fish swimming at 1 body length per second, they have significantly lower baseline cortisol levels. So this is just a summary of the most significant parameters that we found relating to biometry. What we see again was the swimming enhanced growth. We see that fish that were exercised had higher body weight, higher condition factor. Also the heart weights were increased. And we found that the fish swimming at 1 body length per second, they had higher glucose levels. So apparently more mobilized glucose. What we also found, to our surprise, was no significant changes in parameters in the growth axis. On the other hand, what we did find was significantly more deformed fish when swimming 2 body lengths per second. Now, following up that, we performed RNA sequencing of the white muscle to go a little bit into more the growth that we observed. And what we found was many genes involved in, of course, in muscle development and the molecular regulation of those processes, but also in muscle contraction. So we were wondering if there's actually a muscle adaptation going on in response to skeletal abnormalities. And basically, like always in all the exercise studies we do, we find a whole range of immune genes being significantly upregulated. And it could be that the muscle plays an important role in disease prevention, for instance, by the production of myokines. It could also be that exercise causes an inflammatory response in the muscle, like you see in mammals, in order to repair, regenerate, and develop the muscle. So that offers a possible explanation for the effects we observe, because normally you would intuitively say that exercise costs energy and that makes the fish grow better. What we saw in the sea cage was that the fish that were swam at 1 body length per second, they were also more active in the sea cage, at least as determined by accelerations. And they had significantly larger hearts. Now, if you follow that pattern over the hours of the day, you have a reasonable, good idea about the activity fish in the sea cages. So in this case, they were not nocturnal. They woke up at about 6. They show food anticipatory behavior until they are fed at 11. We see in the afternoon, we see a decrease in activity, and it increases again during the evening. So basically, by performing accelerometry, you can see when are these active periods occurring, and it can be very beneficial for timing your feeding events. So to come to conclusions and possible and recommendations, what we would advise is an optimal exercise regime of 1 body length per second on basis of these results. We see enhanced growth. We also see more uniform growth, less chronic stress, more robust fish, at least in terms of larger hearts and having higher condition factors. We also see higher plasma glucose levels, which could suggest that these fish can use carbohydrates better. We see that the fish are more active. They show more accelerations in the sea cage, and the gene expression patterns, they suggest that these fish have an improved muscle structure. They are in relation with development, repair, regeneration capacity. But what we do see, particularly in the fish swimming at 2 body lengths per second, but perhaps there was no significant difference, but perhaps slightly increased when swimming 1 body length per second. And in that case, it may be a good advice to start a little bit later with these exercise regimes in order to avoid that. But that's something also for future research. So all this work is already published in Frontiers of Physiology and Frontiers in Marine Science. So if you want to know more, you can check those papers. But of course, you can always ask me. That's a better way to do it. Just send me an email. Thank you very much.
Thank you. Yep. Okay, so yours are... Put it here.
Yeah. This one. No.
Okay, so we continue, but now we move to see the progress of the work that the PerformFish or sister project is doing, and especially for the work they are doing in key performance indicators, which is, I think, is of interest for the whole audience. So Giovanna, please.
Thank you. Hello, good afternoon, everybody. And I'm Giovanna Marino from Italy, from ISPRA, and I'm going to introduce you to the benchmarking tool based on key performance indicator that we develop in PerformFish project for seabass and seabream aquaculture. We will see very quickly the KPI methodology, how the companies are performing now the benchmarking using the KPI, a short analysis of the KPI at sector level, and the KPI for welfare and the welfare tool that we develop, and some short conclusion. The objective of PerformFish was to provide the Mediterranean marine fish farming sector with a benchmarking system based on key performance indicator to evaluate their performance and to identify source of inefficiency in their production cycle. This was the main objective. It was a request of the 5 producer association and the 20 companies that joined the PerformFish project, and was done before the proposal in 2017, because they really required this system to analyze and compare their performance. The sustainability of food system is a challenge today, more than in 2017. We have the publication of the European Green Deal and the Farm to Fork strategy. And in this day, the European Commission is involved in an initiative inside the Farm to Fork strategy for the sustainability of the food system. So we are in line. The PerformFish KPI system is composed by several components. It was built after a long consultation with the industry, 1 year of consultation to set the more informative KPI. And we start with a list of more than 200 KPI, and at the end we select, we choose 55 KPI that are used in the hatchery, in the pre-grow, and in the grow-out system. These are technical KPI that are used for ranking the biological performance and the technical efficiency. Are a welfare KPI that are used to assess the different welfare condition at different stage. 14 environmental KPI that are used to assess the impact of farming practices on biodiversity, on the use of resources, water, energy, and so on, and 2 economic KPI. In 2017, we started with this job with the company, and at the end, we got a final system to collect in an harmonic way the production data on farm. Then we prepare a manual with standard operating procedures, so everybody can use exactly the same methodology and provide, I mean, not number, but real production data. That is what we are looking for. And then we organize also 10 training courses with producer association and farm manager in Croatia, France, Greece, Italy, and Spain to make the farm manager more confident with the system. This is the dashboard of the PerformFish KPI system. There is a part that is more social and a workspace, and there is a part that is a technical component. Here, very, very quickly, how it is composed. There is a data collection Excel file that are compiled by the farmer and then are uploaded inside a private space and each company has a private space where all the data can be stored. These data are then anonymized and published in one repository that is completely anonymized, and then these data are available for be used by the company using the analytical toolkit to benchmarking and make the analysis and compare the performance. These data also flow into the ISPRA SINA cloud, that is the ISPRA system infrastructure. And here there is a second validation and statistical analysis and sectoral report. These are the statistical analysis, some of the statistical analysis that KPI dashboard provide to the company, so they can see the analysis, some descriptive statistics on each KPI. I mean, the 55 KPI that we are using. They can measure their performance according to a performeter to see how it's going. They can perform some correlation analysis, and they have a matrix that synthesizes their performance. So now we see how it's working very quickly in the hatchery. What is very important is that all the farm manager use the same KPI and collect the data in the same way. So we make this parameterization of the hatchery cycle, starting from 1 day post-hatching larvae up to 2,400 degree days. We use the degree days instead of the day post-hatching because it's more precise. And we consider 3 stocking period, and all these KPI that are the list of 15 KPI that we used is reported by the farm by batch. So we have 5 sampling points in which companies provide data on these 15 KPI. This is just an example of the batch that we have in the system, starting from 2016 up to 2020. You see up is the seabass on the top, and below you have the seabream. And this is the hatchery length of the cycle for seabass and seabream. Now we have for the hatchery around more than 150 batch. And when I say batch, this is a key point. I intend a group of homogeneous larvae that are stocked in 1 or more rearing tanks and managed until the end of the production cycle in the same way. So this is the number of and the size of the batch you can see is quite large. And these larvae are reared in case of seabream only in recirculated system. Instead, for seabass are used also a partially recirculated system, and in few cases open water system. And these larvae, are stocked in different periods, and the analysis that we perform take into account if the hatchery cycle are early, natural, or late hatchery cycle. This is just an overview of some KPI collected during the last phase of the cycle. You see the average body weight, the specific growth rate, the survival estimation, and the discarded fish for spinal deformities in seabass and in seabream. So now I pass to the grow out phase. And in this case also the cycle parameterization is very important. We have different stocking size, different stocking period, and the final size, we have 2 final size, that is in one case is 400 grams, and or in case of farms that prolong the rearing period is 800 grams. The data reporting is at batch level for some KPI and the farm level for other. It's not the same. And the companies report the data at stocking, and then when fish reach 50 grams, and then 150, then 250, and then 400 grams, and at harvest. So we have 6 sampling points during a grow out cycle. Another important point is the localization of the farm. We identify 3 zones that differ for 1 degree. And this zone were identified using 30 years of temperature time series data that were collected by the Copernicus Marine Environmental Monitoring System. Some farms are in zone 1 and have an annual mean temperature less than 19 degrees. Other are between 19 and 20. And in zone 3, we have batch with more than 20 degree temperature. This is the list of KPI that we are using to measure the fish losses, the fish growth, the feed efficiency, and the pension treatment. These KPI are recorded by now for 208 batch and report data from 2016 up to 2021. Some of this batch are stocked in the first quarter and other in the second, third, fourth quarter, and there is a good representation of all the quarter. And this batch also differ per zone. You see, we have zone 1, 2, and 3 with most of the batches, and instead for zone 0, we intend the land-based system. Here is the length of the grow-out cycle in the different zone. You see, it's quite different and more for seabass than for seabream. But this is now I try to report you some of the data that we have elaborated till now from this data set. We closed the data set, the collection of the data in September. So we performed some analysis. I just give you some few examples. And this is for the mortality of seabass during the entire cycle and in the different stocking period. You see that most of mortality are in the first period from stocking up to 50 grams, and then is reduced up to the end of the cycle where there is some more mortality because more than one harvest often in the cage. This is the mortality by period and the mortality cumulated in bass and in bream. You see that mortality is around 12% for bass and around 14% for seabream. These are the losses. The losses are very important because losses is not mortality. At the end of the cycle, the farm collect all the fish, harvest all the fish, and they measure the total losses and what we call the unaccounted losses. So those losses that were not recorded before, and at the end of the cycle, there are some fish that are missing, or in some cases, there are some fish more than expected. And you see that in any case that seabass and seabream company are very good because more than 20% of the batch have a very good analysis of the losses, and the unaccounted losses is quite zero. Other farms, other batch perform less well. This is an important topic, and I just put this slide, only one slide, but we know that this topic is on the floor at European level and everywhere, and is the use of antibiotic and antiparasitic treatment. We see that for 111 batch of seabass and 95 batch of seabream, we have a total number of treatment that is around 73% for antibiotic in seabass and 41% in case of seabream. Is completely the opposite in case of antiparasitic treatment that are higher in seabream as expected than in seabass. But what is more important is that the number of antibiotic treatment per batch is less than one in seabass, and in seabream is near to one. Why is it important? Because you know better than me that we have an organic regulation for bass and bream aquaculture, and the treatments that are allowed according to this regulation are two treatments per cycle. So it means that the companies are now performing better than was expected by organic regulation. So this is really a very good results and outcome for the company. I have, let me say, a lot of possibility to show you some effect that we identify. This is only an example, and this is the growth that is measured with the thermal growth rate, with the JPD and the SGR in seabass and seabream, and you see how the number, the value are different between the zone. So there is an effect important on growth, and the cycle are more longer. This is the FCR and the FCRBO. We know how much is important FCR for the farm because the cost, and also from the environmental point of view because the waste of food and the effect on locally on environment. So these are the mean value, the median value from stocking up to the end of cycle. Now I just show you one DEA example. This is an analysis that used to analyze and compare the efficiency of different farmer batch. In this case, the input KPI are those that you see, and the output is the biomass. And the good news is that that 6 out of 14 farm of seabream have the efficiency that is 1, so this is the top. They are performing very well, and there are other 8 companies that perform less good, but in any case between 0.76% and 90%. So in any case, this is a good efficiency of our company, of our seabass and seabream company. And then the last example is on the last slide is on the welfare KPI. We elaborate a welfare tool that is specifically adapted to seabass and seabream, and this follow the recommendation of the strategic guideline for aquaculture, the new one, that recommend to setting common validated species-specific and auditable fish welfare indicator through the production chain. How it works? It uses the welfare KPI and other operational welfare indicators that are both direct animal-based and indirect operational welfare indicator with the relevance of the different indicators, and then elaborate a welfare index by species. The welfare index that we are using is the adjusted Mazzotta-Pareto index. This is a very powerful tool and is used by the Italian Institute of Statistics for reporting and compare, for make the benchmarking on fair and sustainable well-being in Italy. Okay, thank you, I finished. And so we test this welfare tool in different companies, in different farm, on different batch in bass and bream, about around 50 million of stocked fish. And at the end of this analysis, we see that it discriminates the farm very well and change during the grow out cycle. So it's able to tell about the welfare of fish during farming. For this topic, I invite you to see the poster, the e-poster that was prepared by Tommaso Petocchi and other colleagues of ISPRA, because it's very informative. So the conclusion. We start in 2017. Let me honest, in 2016, because the request of the industry was before the presentation of the proposal of PerformFish project. So are several years that we are playing with this KPI benchmarking system. And today we can say that we have a good suite of species-specific indicators for benchmarking the production performance, for improving the efficiency and reducing the ecological footprint of aquaculture sector. The databases contain more than 400 production batch provided by 20 Mediterranean companies across 28 farming sites. And we have the web tool that the companies are using for benchmarking the performance at batch and farm level. We also developed this welfare score tool that is our intention to test and test again to refine and improve the quality. It's a sort of machine learning system. And in the next year, because PerformFish will last next year, we hope to improve more and more the system. This is the last slide, just to remember that according to the Farm to Fork strategy, sustainable food system will be essential to achieve the climate, biodiversity, and other environmental objectives of the Green Deal, while improving the income of primary producer and reinforcing the European competitiveness, especially in aquaculture, because we know that it's not so good. So this system, the PerformFish KPI benchmarking system, offer a common and comprehensive approach to address sustainability issue in Mediterranean aquaculture and to report on the uptake of sustainability production practices by aquaculture primary producer. That is what the Farm to Fork strategy requests. Thank you.
Good. Thank you very much, Giovanna. We move to genetics. Giovanna, this is yours. Welcome. Okay.
Thank you. Hello, everybody. Welcome to work package 3. It's about genetics. My name is Anna Sonesson. I work for Nofima in Norway. And other partners in work package 3 have been 2 industry partners, Galaxidi from Greece and Colmarex from Spain. And other research partners are Inya from Spain, Roslin from the UK, Ege University in Turkey, and Wageningen University. Here is the content lists. I would like to start with a small introduction to selective breeding. So the FAO came in 2019 with a state of the world for aquatic genetic resources, a report which shows that we have more than 600 species in culture in the world. Aquaculture species, 40% of all aquaculture species are produced from unimproved wild types. So we see there is a big gap for the selective breeding community to fill. The report also states that 45% of reporting countries note that genetic improvement is still not significantly impacting aquaculture production in the countries. And this is quite strange, because selective breeding is a very powerful method to increase productivity and disease resistance, etc. So there was a review done quite some years ago on the effectiveness and how much gain we actually get after 1 generation of selection. And this review showed an average of 12% per generation for growth. And we know today with the current molecular tools that we can get up to 25 and 50% more, and in theory up to 100% more gain if we have genomic information using genomic selection. And there are also studies that show the economic impact of selective breeding. So it's about 0.2 euro per kilogram fish has been shown both in gilt-head seabream and recently in Atlantic salmon. So what about our 2 species of the MedAID project? The European seabass has breeding programs, and in 2016, when we started the project, it was estimated that about 50% of the farmed seabass came from selective breeding programs. The gilt-head seabream was not that well developed, But there were also commercial breeding programs for seabream, started in the early 2000. A couple of words about a genomic tool that is very, very essential for our work. It is the SNP array. It's an essential tool for all genetic studies, basically. So we are then thinking about genomic parameter estimation, gene mapping, genomic selection, co-ancestry measurements, and population genetic study. And when we started the MedAID project, we knew that there were SNP chips with several tens of thousands of biallelic markers for Atlantic salmon, trout, and later we have seen development of a carp, catfish, shrimp, and tilapia. But there was at that time no SNP array for our two species. But we had a database of SNP markers from previous projects. So one of the very important objective of work package 3 was actually to develop this combined SNP array for seabream and seabass. And then we were using this for different applications. One was to test them for key production traits in seabream and seabass breeding, to discover genes for these production traits in seabass and seabream, to characterize the genetic diversity of populations of farmed and wild seabass and seabream, and to test measures of inbreeding. And finally, we have used this SNP chip to assess the existence of extent for G-by interaction for temperature on production efficiency in seabream. So Ross Houston, he will present the first objective on the SNP chip, and John Bastiansen will present the work done on the G-by interaction, and I will have very short overview of the other objectives here. First, a little bit about the lipid traits that we chose to work on in the seabass and seabream. Why did we do that? Yeah, first is that lipids are important traits. They are linked to production efficiency, health, and product quality. And excess dietary lipids that are not deposited in the edible muscle or used as energy source for growth are considered a loss and an indicator of low efficiency. We don't want to have lots of visceral fat in the body. We know that excessive lipid deposition in and around internal organs increase the risk of metabolic disorders, oxidative stress, and they give inflammation. And a last reason for looking at lipid traits is that we know that omega-3 fatty acids are important for both human and fish health. So here are some very brief results from the work done in seabream. So we can conclude that selective breeding is a promising tool for increasing EPA and DHA, two important fatty acids, omega-3 fatty acids. They had the heritability of 0.39 and 0.34 in our data set here. But we don't know the underlying mechanism for getting this genetic variation. It can be linked to degree of deposition, beta oxidation, or the omega-3 bioconversion. The total muscle fat content, which has a trait with a high heritability of 0.33, but the correlation to body weight is quite low. So we can conclude in this study that the total muscle fat content and the body weight seem to be genetically independent traits. The same is for body weight or growth and percentage EPA and DHA. What is different to Atlantic salmon that we have studied a lot before, in seabream we see that fatty acids 16(1) and 7, which is a marker for de novo lipogenesis, had a very high heritability of almost 0.5 in this data. And finally, we saw genomic prediction accuracies of 0.58 to 71 for these lipid traits in green. For the seabass, it's a bit different. It's not exactly the same fish when it comes to lipids. Here we saw different heritabilities for DHA and EPA. That's what we also see in salmon. So DHA has the high heritability and EPA has a lower, 51 and 16%. But also in this species, we saw total muscle fat content had a high heritability. But in bass, we also had data on liver fat that we know is like a metabolic important organ for fish, just like in humans. That had a lower heritability of 17% fat content in the liver. We saw in the seabass that EPA and DHA had a very high genetic correlation. It was close to 1. Of course, we have errors in these estimates here, but that's also different than we see in the other species we have looked at. And we saw a low correlation between muscle fat and body weight, also here in this species. And again, we saw quite all right prediction accuracies for genomic selection of 0.7 and almost 0.8. Then I'd like to jump over to the, one more word about this. So there will be a presentation on the BREAM data in the selective breeding session tomorrow. Then I jump to the population genetics. This is also a topic that will be presented in the selective breeding session. But why are we interested in population genetics? Yeah, we need some information about the population genetics when we want to set up a base population. Because we know that the genetic variability in the base population determines subsequent genetic progress. The base population can be created from wild or found populations, and it's necessary to determine the relatedness within and between populations when we take them in to breed them. We need to know, we need to have a good genetic management of an ongoing breeding program. So for that, we need to know ways to control inbreeding and calculate effective population size. And we want to do population genetic studies to study farm and wild fish interaction. So the data we had here was collected together with the PerformFish project. So this is these are the sites for the wild populations. In addition, there were farm populations. And the overall conclusion is that the wild and the farm population had differed genetically. So on the left side, you have the seabream, and to the right, the seabass. And the upper panels show the wild populations, and the lower show the farm populations. And as you can see in both species, the wild populations are more homogeneous. And in the seabass, you see there are one extreme, I think it's Morocco and Cyprus, but for the rest, they are also quite homogeneous over the whole Mediterranean area. So that's the wild at the upper, but at the lower, you see much more heterogeneity. What that study also found was an effective population size for the wild population was more than 1,000, which is very good. But for the farm population, it was lower than the suggested effective population size of 50 to 100 that you want to have in a sustainable breeding program. So that means for the farm population, we need to have selection and mating strategies that account for the inbreeding. And because the wild and the farm populations are genetically differently, we must avoid escapees that could have undesirable genetic effects in the native populations. Now I come to the overall impacts of the work package 3 of MedAID. Maybe the most important impact is that we developed and validated a SNP array for seabream and seabass for use in genomic selection, but also for the other aspects. But for the industry, it's, I think, mainly for that purpose that it was useful. We have evaluated lipid-related traits in elite populations of seabream and seabass. They're important for the health, production efficiency, and product quality. The low effective population sizes in many farm populations show the need for improved management of genetic variation in these populations. And the results also show that we have to be careful and avoid escapees that could have undesirable genetic effect in the native or in the wild populations. And finally, that you will hear later, the WP3 also shows that there is genetic bioenvironmental effects for growth traits between West and East Mediterranean. So we need to have local adaptations for their breeding program. Thank you.
Okay, now it's Ross. Hi, Ross. Do you see yours? No. This. Open Drive, because I don't see it. I've got it on my laptop. Oh, but you have Open Drive.
No, it's
But
on my
you have-
laptop.
Can
Yeah,
I,
you can put it on.
and then
yeah.
we move
We
to,
can put John first if you want. Yeah.
okay. Okay, we are going to change John, please. Mhm.
Thank you, Bernardo. I'm going to talk, as Anna also already introduced, about the experiment that was done, which was also referred to by Alicia, but the part that we analyzed was to look at genotype by environment interaction. And I'm like this, okay. So we looked at the interaction of the genotypes with the environment in the two commercial sites in Spain and in Greece for gilthead seabream. I'm going to first show how we designed it, so what the rationale was, then the data collection that was specific for the G/E analysis, the results, of course, and then some recommendations. The environment is there are many environmental parameters. So the one environmental parameter that we try to target is temperature differences. And that's why the sites that were used were chosen to be in the east and the west of the Mediterranean. So as explained before, this was a batch of seabream from a single spawning event from a group of parents. And around 80,000 of them were transported to Spain, and another 80,000 were put in a sea cage in Greece. And they were grown to a harvest size of around 400, 450 grams. And at that point, we came in and we harvested, or some of the fish were harvested and measured for growth and quality traits. So we did measure around 1,000 fish, both in Greece and in Spain. And in order to use those individual measurements, we need to know their relationships. And since they were not, they were from a mass spawning event, so we needed to genotype them to identify their genetic relationships. So that was done with the array that was developed within the Mediterranean and PerformFish projects. The results that I'm going to show are for these production traits that are listed here: harvest weight, fillet weight, viscera weight, and also the fillet fat percentage. We also measured heart weight, liver weight, and some other measures on these individuals. But I don't include them here for the interest of time. They are in the paper that is coming out in aquaculture very soon. It was just accepted. So we looked at genetic correlation. So what we were after is, do these families or these parents of the fish that were measured both in Spain and in Greece, do they rank the same based on the data from Greece as they would based on the data from Spain? So if you look at this cartoon on the right, there's some on the left of this cartoon, there is the genetic estimates of individuals in the, let's say, cold environment, and on the right is the genetic estimates of those same parents based on their offspring that grew in the warm environment. And you see that there is some re-ranking going on. So if you look at these two fish, let's say the green fish, it is near the top in the cold environment. It's also near the top in the warm environment, but there are quite a few better fish that you could select in that warm environment. The same for the red fish. It is very close to the top in the warm environment, and it's a little bit down the list in the cold environment. And remember that these are the estimates of the genetic potential of these fish. Of course, we'd like to have fish that are all the way there at the top, but there's only one. And we need, for the breeding program, we need to maintain genetic diversity, so we need to go down this list a little bit. So what is happening then if you would select in the warm environment? So if your breeding program is in the warm environment, you might select this range of fish at the top. And then if you look at those same fish, their average performance in the cold environment, it ranges, I indicate in green what the range is, and also the average performance of those fish that you would select in the warm environment is slightly lower. It's still an improvement, but they are lower than what you would have expected based on their superiority in the warm environment. So this genotype by environment, we typically measure this as the genetic correlation. Now the cartoon I showed is for a correlation of 0.7. So correlations range from -1 to +1, as you all know. And what we estimated for the growth rates, harvest weight, fillet weight, and viscera, is that these correlations are in the range of 0.5, 0.45 to 0.6. So that means that there is a significant re-ranking between the environments. For fillet fat at the bottom, we see quite high, close to 0.9 genetic correlations, which means that if you would select in one environment on fillet fat percentage, you would expect to get a similar response in fish that are growing in the other environment. So what can we conclude? Because of these relatively low values, around 0.5, that there is strong G/E for these production traits, harvest weight, fillet weight, and growth rate, for gillhead seabream between the east and the west Mediterranean side, and that it is very limited for fillet fat percentage. Recommendations from this work is that, so we did the genetic analysis based on the genotypes with the SNP array, showing that it is suitable for this type of analysis. We recommend that producers benefit the most if they source their fry from a breeding program that is targeting their local conditions. And so here we looked at temperature differences east and west, but of course, in time, environmental conditions are also expected to change. So there's a need for breeding programs to take these changing environmental conditions into account as well. Thank you to the two companies especially, and a lot of people that contributed. I want to highlight one person, Behnam Gulzari, who's a PhD candidate in our group, who did the majority of the analysis and the writing of the paper. Thank you.
Let's try this side.
Okay, thank you, Bernardo, and sorry about the technical issues there. So I'm going to give a quick overview of the snip array, the development and applications of the MedFish, seabass and seabream snip array. I'm up here presenting, but I want to pay particular thanks to Carolina Peñalosa from MedAID, and also Teresa Costas and Luca from PerformFish for major contributions to this work. So just briefly, Anna's given us a very good overview of the need for selective breeding, what it can offer, and it has major potential. And also that genomic selection is really where the state of the art is in advanced breeding. And that's important because it can give us improved accuracy, which leads to faster and more sustainable genetic gain with reduced amounts of inbreeding. But of course, genomic selection requires this development of a high throughput genotyping tool. And so our aim was to develop this combined species snip array with 60,000 genetic markers. The reason for combined species is because the cost per unit is lower when you order a higher number of units. So we're able to order more to tackle both studies for seabream and seabass. So how did we do that? Well, it was, as I mentioned, a very good collaboration between PerformFish and MedAID. We sampled 27 populations of seabream and 24 populations of seabass, thanks to a huge array of collaborators I'll mention at the end. And you can see here on the map where the samples were taken from for the two species. The workflow for the sequencing and discovery of the SNPs in the two species is depicted here. So DNA extraction was performed from the populations. The DNA from a particular population was then pooled, and they were sequenced in pools, high throughput sequencing. This was then used for SNP discovery in the pools using fairly standard bioinformatics techniques, giving us a preliminary list of SNPs, which were then filtered down to the final list that were put onto the array. So you can see here for both species, we generated initially a very, very large number of SNPs, about 34 million in bream and 17 million in seabass, and we went through various stages of filtering to end up with a SNP array that had approximately 30,000 for each species that were fairly evenly distributed across the genomes of those species. And you can see there on the right the distance between markers, approximately 16, 17 kilobases. And this depicts the distribution of the SNPs on the genomes of those species. So track A in the plots are the chromosomes of the species. Track B is the known nucleotide diversity, and track C, we tried to mirror the nucleotide diversity with the distribution of the SNPs on the array. We then tested the SNP array performance in diverse populations, including the populations that we used to discover the SNPs. We categorized them according to their genotype and performance, and the results were very, very good. So there was approximately 90% conversion from a theoretical SNP discovered by sequencing to a high-performing, high-quality polymorphic assay on the array, which is amongst the best-performing aquaculture SNP arrays, which is fantastic. We've already heard about some applications of the array, but just to name a couple more. So we did some initial testing of the array performance in diverse seabass and seabream populations to look at its utility for detecting population structure. And you can see from these PCA plots here that we were able to pick out population structure in the samples. But to find out more, please see the presentation of Maria at 3:30 p.m. on Thursday. And just to sum up, so we created a combined species 60K SNP array and validated it in multiple seabass and seabream populations. The work has been published in the journal Genomics. We have an extremely large database of SNPs from characterizing the genetic diversity of seabass and seabream. These are publicly available. There was a high conversion rate. It's been shown to be informative for diverse farmed and wild populations. Useful for detecting population structure. And two other presentations, one of which was supposed to come immediately after this, but actually came before, is John's, and the other is that I noted in the program is, as Anna was talking about, it's 5, 10 and Wednesday, and I'll talk about genetic parameters and GWAS of lipid traits. Okay, thank you very much.
Okay, thank you very much, Ross. We move to work package 4 on health and welfare. Let's see. Which one? It is this one? No.
Final event. Oh, it's not.
Ah, no, this is Polanco. Ah. Mierda. This? No.
No, there should
No,
be a
no,
date
no.
behind it.
Package 2. Ah, okay, here it is. Sorry.
Yes.
Yeah.
That's the one.
Okay.
It's just to use this.
Yes.
Okay, everybody, good afternoon, and happy to see that some are still hanging on. It's getting late, but this is the last work package, so there you have to hang on to 3 more presentations. On the other hand, we are talking about work package 4 on health management, diseases, and welfare. And I like to say that if we don't take care of at least infectious diseases and keep the fish happy, there will be no growth and there might be no industrial aquaculture at all. So this is the foundation for what we are working with. We have 9 partners in this work package from Spain, Norway, Croatia, Denmark, Egypt, France, Italy, Tunisia. And we also had a subcontractor from Greece. So we had the whole of Mediterranean represented in this work package. I go straight to general objectives. I also, before I do that, I'd like to say thank you to all of you who participated. And I do not have a full overview of everybody, so none mentioned, none forgotten, but I'm very appreciative of the effort and the enthusiasm that was shown in this work package. The work package has 4 objectives. One is here to develop or to use risk assessment tools to look at emergent pathogens in the Mediterranean. We know that there are not many, let's call it serious infectious diseases. So that's why there is a good starting point, and we need to be well prepared for infectious diseases that might arrive in the Mediterranean. Also, we like to focus in this work package on communication. It's very important that we communicate and use the same terminology so we understand each other. As you know more than me, that the Mediterranean is surrounded by, I don't know, 20 countries, some EU countries, some none, with a variety of languages and traditions. So this is a very important objective to establish a common communication platform. And this also goes for harmonization of diagnostics, because there are trades going all across the Mediterranean, and we need to understand each other, and we also need to use the same methodology, so that when a lab in Spain finds something, it's understood in Egypt and vice versa. We have some diseases in the Mediterranean, and the VNN is one of the more important. And in this objective was also to develop preventive tools to control the disease better than today. And welfare, as I mentioned, without good welfare, there will be no aquaculture. And welfare, there's nobody buying the products if the fish isn't treated according to standards. So this is a growing area of interest, and it needs to be addressed very thoroughly. I go through the various tasks and aims, and the first one is divided in two. First, we wanted to use a risk assessment platform to look at the risk of emerging pathogens or relevant pathogens entering into the industry in Mediterranean. And as an example, we used VNN, as was identified as the more important disease to control. The second task in this under this objective was to look at biosecurity measures on specifically on farm levels and try to quantify biosecurity. We all talk about biosecurity, and the COVID situation is a very good example. It's never been easier to talk about biosecurity and epidemiology before then, the last 1.5 years, and everybody understands the importance of it. But we have a lot of measures. Some are important, or you could say all are important, but some are more important than others. And trying to quantify this is a very interesting exercise, and it's also important for farmers to spend money on measures that really matters. So for 1A, we saw that the purchase of fish is definitely the most risky activity going on in the industry. So there is a huge need to try to identify and reduce or eliminate what we call introductory risk or external biosecurity. Focus on external biosecurity. And we also saw from the risk assessment that there is a need to use that tool in order to try to identify the various factors and their importance. And it's interesting to see, if you see the map, this is just to illustrate that there is a trade going on all across the Mediterranean, from east to west, from north to south, and with all this variety of lands or countries, no one better than the other, but there's a huge connection between all countries in the Mediterranean. And even though how big Mediterranean might be, it is a pool that everybody is playing in. So horizontal transmission is very important, and all these trades show how important it is. So I just try to look in this COVID again, that there needs to be some knowledge or some structure that can control the trade in a proper biosecurity way. And then again comes this communication harmonizations in diagnostics. For 4.1, Soraya Tavon-Panic will talk a little bit more about that. For task 4.2, it was to strengthen diagnostic capacity by harmonizing competencies. And again, just we tried to establish this network for communication. We had a forum, internet forum that we established, and it worked for a while, but we all know that this is a complicated thing. Again, look at to improve harmonization on labs, capacity building. We all need to have sufficient capacity in the various countries, or at least around the Mediterranean. In order to look at this competence, we organized proficiency testing, specifically on VNN, and we organized training activities. So we saw that there is a need for capacity or improving the diagnostic capability along the coastline of the Mediterranean. There are lots of techniques that are lacking, and we need to look into it in order to be sufficiently secure that when you have a diagnosis, it is properly validated or properly done and validated. We also saw that the forum we established is very important in order to keep a dialogue between stakeholders, I just use that word for everybody, within the industry. But as I said, it's hard to keep these platforms. So webinars, which have become very popular the last year or so, that could be a more efficient and a more flexible way of keeping that communication. And again, with the labs, there is a need for diagnostic methodologies, a continuous training so that all labs engaging in the industry are at a sufficient level of competence. And in this task, we developed a manual or a book of diagnostic methodology, which I could say should be the Bible for all labs around the ocean, or at least it's a draft, so we can always make it more perfect. But here it is, so you can all go on the website and download it, and it's well worth doing. Task 3 was looking at the notavirus and look into these virus-like particles to use them for vaccination. And this also will be presented by Anna Toffen later today as the second talk. Task 4 is on welfare indicators, OVIs, or profiles. And we look primarily on seabream because seabass was focused on in PerformFish. And the aim for this was to list OVIs that could be used to validate fish welfare, focusing on animal-based indicators. And we have this list. It's a theoretical based list with welfare that can be used to start working in order to establish some welfare certification scheme. We see that we need to put more effort into supporting or to understand the OVIs, and also, of course, validate them with the industry. I took this illustration because we are handling fish and we are treating fish quite bad. Maybe because we cannot read its expression, we only have a live or a dead fish, more or less. So in this work, it's important to start to understand the signals that our farmed animals in water expresses. So this is a very important task to try to be more sophisticated when it comes to understanding the need and the welfare of these animals. And I think that is also something very important for future and the perception of aquaculture. So that was what I had to say. Thank you.
So, hello everyone, my name is Saraya Thawanpanish. I'm a senior researcher at Norwegian Veterinary Institute. So the talk I will present today is part of MedAID Work Package 4, Task 4.1 that Edgar mentioned earlier. The aim is to develop a generic system for quantification of biosecurity measure for seabass and seabream productions. And even though only my name appear here, the work is the team effort from all our work package partner and our subcontractor. So biosecurity is defined by the OIE as the set of management and physical measures that designed to minimize the risks of pathogens introduction, establishment, and spread in aquatic animal productions. So being able to measure is being able to improve. Measuring biosecurity provides us a way to pinpoint action that apply and identify weakness and gaps and improve the farm overall biosecurity. So for this task, a risk-based weight scoring system was developed for Mediterranean sea bass and seabream farm using the method created by Ghent University called BioCheck, which is the system that approaches biosecurity in a general manner and focusing on the transmission route shared by numerous different types of infectious agents. The first step of this task involved discussion with subject experts, including fish health specialists, veterinarian biologists, and epidemiologists. And we review literature, existing documents to gain a good understanding of the current production system and lay out the sequential production flow throughout the production system from hatchery, pre-growing, and ongoing facilities. As well as we identify transmission routes and action that have potential for disease introduction and spread. And in order for us to collect information related about biosecurity practice on farm, we developed a questionnaire and distributed to seabass and seabream farm in the Mediterranean. The questionnaire contain a total of about 160 self-explanatory questions that were structured into 19 sections in order to get the data on the farm general characteristics, production management, health management, disease reporting, diagnostic capacity, biosecurity practice, and record keeping, and so on the farm. And the questionnaire administration was done using in-person interview by our partner and subcontractor. We use a follow-up phone call if they are unclear of the answer or the question that needed. There were 88 farms from 8 different countries that participated in this questionnaire survey. And these farms were grouped based on their production types and location to hatchery, pre-growing, and on-growing facilities. And we used stepwise approach for developing the biosecurity scoring system. By using the data collected from the questionnaire, a selected subset of questions were pooling and used to form a new dataset consisting of questions that relevant to external and internal biosecurity. The external biosecurities consisted of questions that can be grouped into 7 different subcategories, which is listed here in the blue color font. And internal biosecurity also consisted of 7 subcategories that are listed here in the green color font. And each subcategory contains relevant number of questions, ranging from 4 to 12 questions per subcategories. Answer to the question were assigned a score from 0 to 1. So farmers were scored depending on how they answer the different questions. For example, the score 0 indicate a total absence of biosecurity measures or full presence of risks, and the score 1 indicate that there is a full presence of biosecurity measure or the total absence of risks. Yeah. And not all biosecurity measures are equally important depending on the pathogens. And because of the disease transmission pathway have different efficiency to introduce and spread depending on the pathogen. Therefore, the score is multiplied by weight factors. And to get the weight factors, we require help from subject experts. So the experts were asked to give a weight to each questions and also allocate 100 points divided between subcategories. And this table shows the expert weight for VNN. Then The score for the subcategory and category score were calculated as present here in these two equations. The overall biosecurity score is then calculated as the mean of these two main categories, external and internal biosecurity. And these slides present the average score of the 19 participated hatchery. The average external biosecurity score was 71%, and the average internal biosecurity score was 74%. And among the external biosecurity, the subcategory with the highest score was biosecurity that connected with personnel and visitor, whereas the subcategory with the lowest average score for external biosecurity was location of the farm, which related to the example of how many or what production type and how close the neighboring farms are to the farm being evaluated. And for the internal biosecurity category, the subcategory with the highest score was feed management, and disease management had the lowest score. Okay, so in conclusion, this is the major constraint in aquaculture, and biosecurity is critical for sustainable development of aquaculture. So this work emphasizes how biosecurity measures and their relative importance can be quantified and documented in an objective way. And the developed quantitative biosecurity system helps to identify gaps and weakness in the actual biosecurity plan. This system assists farmers to allocate resources and tailor the biosecurity program to fit the risk profile of the specific farm. So, and if the system is applied in a region, it also helps to compare a specific farm biosecurity with the average biosecurity score obtained by the neighboring farm, so that the owner can benchmark their biosecurity and evaluate the risk profile of the regions. And this benchmarking may give owner the impelling reason to improve their farm biosecurity. Oh, sorry. I haven't finished, but so for the future perspectives, this system can be modified to fit various farm characteristics for different exposure and for different disease agents. So at the moment, the method developed here from this MedAID project has been applied, and at the moment is in the progress of being used, the methodology to test applicability of this system for Norwegian salmon productions. And these tools is aimed for farmer self-assessment tool, being developed to be a farmer self-assessment tool with the user-friendly automated dashboard containing the functionality so that the farmer interested in the objective evaluation of farms' original biosecurity can have a secure access of their own information. That's all. Thank you for your attention. Yeah.
Okay, thank you, Saraya. Let's go to the last presentation. Here it is.
Okay. Good evening, everybody. Thank you. I'm honored to be the last one, and also have the responsibility to be short because I know that you are all tired. So my name is Anna Toffan. I came from Italy. I work at the ISPRA, and together with my colleague in DTU Aqua in Copenhagen, we work together on the VLP's vaccination against viral nervous necrosis. The viral nervous necrosis, we already saw, is the most important viral disease in Mediterranean farming. So the VLP stands for virus-like particle, which are structures that possess the same antigenic properties and size of the real virus. So the fish immune system recognizes this particle as a live virus and react to them. But this virus-like particles are not infectious. They cannot replicate, so they are completely safe. They can produce in yeast, so they can be produced in high yield with a relatively low cost. And the prototype of these virus-like particles were developed during a previous European project called Target Fish, and they are now produced by a company called W42. During the 4 years of the project MedAID, we tested in experimental conditions this VLP as a vaccine, and we performed 6 experimental infections in 2 different experimental aquaria. The first one was in DTU in Copenhagen, and the second one was in Italy at my institute. We tested thousands of fish of different sizes, ranging from 5 to 90 grams. So our results are pretty robust. We perform all these experimental infections in order to characterize the safety, the potency, to evaluate the long-term protection, the serological antibody response, and the immune gene response stimulated by this virus-like particle vaccine. This is how the experiment on average were performed. So we vaccinated the seabass by intraperitoneal injection only once. We selected the single administration protocol. We tested different doses with and without the adjuvant. And then after the vaccination, we collect organs and blood in order to characterize the antibody response and the immune system response of the fish. Then we challenge the fish at different time point, post-vaccination, from 2 months post-vaccination towards 8 months post-vaccination. And the challenge was performed both by buzz or by injection, depending on the experimental infection. Then again, we collect the organs and blood in order to characterize the pathogenesis of the disease in vaccinated and unvaccinated fish. Finally, we calculate the cumulative mortality, and we perform statistical analysis like the RPS calculation, Kaplan-Meier survivors, and so on. And this is important to say that in each experiment, we added a negative control group, of course, and also a commercial, a group vaccinated with a commercially available product in order to compare. Then, for when possible, and in many experiments, we recorded the weight, and we also replicate tanks. And in some cases, I don't know if you can appreciate in the picture on the top right, we identify the fish singularly in order to follow the single fish throughout the experiment. It is quite hard to condensate all the results we obtain. We try to put all the results we have in this graph. We plotted the survival against the time post-vaccination expressed as degree days, and the arrow showed the different time of challenge. So we could observe dose-dependent response and a time-dependent response, but importantly, in blue line, there is the higher dose we use, is 40 micrograms. I don't know if you can read. It's 40 micrograms per fish, and this dose produce a high survival for a long time, even after above 8 months post-vaccination. So this was a real good product. So we want to understand why this virus-like particle vaccine works so good. And we checked the serological response, and we discovered this is the result of a single experiment, but this was consistent throughout the experiment. We discovered that this virus-like particle stimulate the humoral response in a very strong way, compared, of course, to the negative control, and even also compared to the commercial available product. And the most important thing is that this antibody response was detectable by ELISA, which is a commonly test used for the detection of antibody, but also through the seroneutralization test. The seroneutralization test is a less used test because it's quite time consuming, but it is the only test capable to recognize those antibodies really protective for the fish. So we observe that our vaccine stimulate particularly the production of neutralizing antibodies. But this was not the only probably reason why this vaccine worked so good, because we perform gene expression days. These pictures are just some examples from the 7-day post-vaccination, and we could observe in the green line that the VLPs, vaccinated fish, had a distinct expression profile compared to the other non-vaccinated and commercially vaccinated fish. So we think that the VLPs triggered immune response in a better way than the other product we tested. So to conclude, Our DLPs-based vaccine did not induce vaccine-related death. I didn't say, but it is a completely safe product. And it did not impact the weight gain over a period of 8.5 months. So this is really important from a zootechnical point of view. Our vaccine does not need for adjuvants. We tested with and without the adjuvant. The adjuvant didn't improve the performance of the vaccine, so we prefer to avoid the adjuvants to escape from and to avoid the side effect related to the use of the adjuvant. The VLP's doses ranging from 20 to 40 micrograms per fish provide a significant protection, meaning above 80 of RPS in experimental challenge after a single intraperitoneal injection. The vaccine induced a high specific and long-lasting antibodies response, as I told you before, detectable both by ELISA, but also by seroneutralization test. And the VLP's vaccine provide protection in experimental infection up to 7.5 months post-vaccination. And the survival was really high, 90% of the vaccinated group survived. So we think that this vaccine efficiently stimulates both innate and adaptive immune mechanism in a well-balanced pattern. And that is why it works so good. And so we are really happy with this prototype of vaccine. Thank you.
Okay, thank you, Anna. Thank you very much for your contribution. So this has been a long session. I propose that we give the chance to those that have or may have doubts or questions to make them. It's not going to last for a long time, but let's see, at least give you the opportunity to ask if you have doubts about all these presentations that have been presented this afternoon. I don't know, Cristóbal, have you looked at the?
Giovanna, one question. The benchmarking tool that you are presented in the PerformFish is or will be available for all companies in the Mediterranean, or just belong to the partner to the PerformFish, or will be open to other countries, not European, inside your consortium, like Turkey, like Egypt, whatever?
This is a political question instead of technical question. I'm not the coordinator of the project. So we hope, I hope, personally, I hope that now that the system is well running, next year, you know, in PerformFish we have 1 year more. Next year it will be open to all the Mediterranean, because it's very interesting for us get also batch and farm data coming from different locations across the Mediterranean. I mean, Turkey, Tunisia, and Morocco, and so on. So I'm confident that the system will be open to all the companies. Now it's ready, so run for 10 companies or 100 companies is the same effort.
Well, a very important comment. I don't know how many producers are here in the room, but it's really important. I think that in our experience, one of the best things that MedAID may have contributed to the sector, and I suppose that PerformFish too, is that when performing the surveys, the questionnaires, there was a very good interaction with the producers. Just only by performing those surveys, those questionnaires, is a way of learning, self-learning, what Soraya has presented about the questionnaires for the risk assessment, the biosecurity. It was also a very good exercise for the companies to audit themselves and to check their biosecurities by their own. The work I have seen that PerformFish is developing the standardization for the collection of information for the key performance indicator is also very, very important, because companies, they collect the data in different ways. So to advance on that, on that way is very important. So I congratulate you for that. We hope that the data will be available for the community. I don't know how you are going to deal with that, but as much as you can do for share that data will be very appreciated.
Bernardo, just to inform you that we are going to prepare the third reporting period for the Commission. And in this reporting period, we will provide the data on the performance of seabass and seabream companies. This is a deliverable for us, and we will report all the key performance indicator and all the data that we collect. I mean, in anonymous way, but in any case, we will have a picture of the sector. I think a quite good picture of the sector.
Okay.
If I may, as representing the European Commission funding this money with public money, I can remind everyone the rules of the game. This is public money, but not public money just to do research and to keep the results in your drawer. There is every single beneficiary, any single participant in the project has signed a contract. In the contract, there is a very clear article on the obligation to do communication, dissemination, and exploitation at the end of the project. So we will not allow that the results just remain there. The results have to be exploited. Then this is research. So within the project, someone has developed the knowledge or a particular piece of work or a particular device. Anything has been developed by someone, and this someone is the intellectual owner, has the intellectual property of these results. So, and then every project has a consortium agreement internal. The Commission doesn't belong to this consortium agreement. And here there should be a clarification on what belongs to who and who is going to exploit what. Exploitation means not only, well, ideally is to bring to the market and to make money, but exploitation is also transferring of knowledge, using this knowledge to do further research, but definitely this should be exploited. And then on the data, Bernardo was saying about making the data open. In Horizon 2020 and now in the new program, Horizon Europe, there is this policy of open science on publication that should be in a repository open to the public. That's compulsory, and data is compulsory in Horizon Europe. It was optional in Horizon 2020, but I think the two projects are participating, and you should make a way to make the data available for other people to use the data, and not to need further funding to generate the same data to do further research. Okay, only that. Sorry.
Just a very short replay. There is no owner of this data. The data belongs to the companies that provide the data. And they provide the data because the system is anonymous. So they can compare their performance without showing to the other companies how they perform. According to the consortium agreement, now we have the possibility for me, the opportunity for me to finally show this data. But let me say that I received the data set, the whole data set early this September. So I conceived the system, but I am not the owner. They use the system, and now finally the system is well running, is well going. And now the data for the first time flow from the anonymous system to the ISPRA SINANET, the ISPRA cloud, where we can make this first analysis that I present today. But for December, we have to report a public deliverable with the data for the Commission and all the other stakeholders that are in some way involved and in aquaculture and seabass and seabream aquaculture.
Well, anyway, we encourage you to do this very hard work that we are also doing. It's not easy to, I mean, today they got the data from the companies to keep the confidentiality and to deal with both issues at the same time. Sometimes it calls my attention that we think that the confidentiality that the companies claim is excessive, because many times you can get similar information from other sources. But anyway, this is what's going on. Hopefully with these exercises that we are doing, we will have to change that mind that not all producers, but many producers they have, because without the information and without sharing information, it is difficult to take decisions, or we will take decisions late, which is also no good.
Just briefly and for clarification, you are talking about background data, data brought to the project. Consortium agreement should clarify also who is bringing what and for what purpose for the implementation of the project, and what happened if after the project this data should be still available for further use or not. But the rule on the open data is data generated in the project. I understand, and I knew your concern about the confidential data from the companies, but this rule, I say that the open data is a general rule, but it's on the data produced by the project. So it's just not to mix data. You were talking about the data brought to the project, background, and I was talking about foreground or results data, just to clarify. Yeah. Thank you.
Just wanted to give you one example of the difficulties that the researchers have. You have seen the last presentation from Anna Tofa about the results of the bat signs. You may have seen or pay notice of one of the slides showing the big difference of the protection got with the bat sign of they are developing compared with a commercial bat sign. So that is also a difficulty that we are facing for the dissemination of the results to ensure that what is going to be published is completely right, that the publication of those results before to being disseminated pass the needed evaluation and the needed filter before going disseminated, because it may have commercial implications. So we are dealing with these issues and paying attention to them. You say that Alicia is leaving.
I mean, the other person, Alicia has to leave immediately. Yeah. If there's any quick question, and for any other of the presenters, please.
No. I don't know if does anyone have any question for today's session? No. Well, one of the things that just to end, because we are ending here, this final event has been a good opportunity for us to interact, researchers, partners from different countries and from different disciplines. That has brought us opportunities to develop things that otherwise could be difficult, like the benchmarking tool, the work that we have done with the colleagues from Cantabria, this very huge work we did to collect information and samples all over the Mediterranean. In MedAID, we counted with the participation and collaboration of countries from the south of the Mediterranean. They have been very open, and I might say that probably more open than Europeans want. So we thank you for that, and we hope that you don't change, at least in those aspects. Well, I just wanted to put that on the table. I don't know if there are more questions or comments.
No question in the chat. It's just congratulations for the season.
I'm sorry, no question in the chat. It's just, well, so many people are congratulate us for the presentation and thank you for sharing.
What you have seen today, what you have followed is the results of all the work packages, except the deliverables that we are finishing now, which are those deliverables related with the work package 8, which is the integration of all the work. But you have seen, you have listened most of the components. So what those deliverables are going to produce is not going to be very different of what you have already been able to follow. That information, as we were saying at the beginning, is going to be uploaded, made available for everybody in the MedAID toolbox in a friendly way, in a more friendly way that the MedAID web page that we have for the purposes.